let's go ahead and introduce our SGA college-wide team.
Good afternoon, everyone. My name is Olivia Assing-Nunes, and I'm the East Campus Student Government President, and I got vaccinated to keep myself and others safe.
Hi, everyone. My name is Abigail Watts, and I got vaccinated. I'm the SGA president of the Lake Nona campus, and I got vaccinated because members of my family are high risk.
Hey everyone, thanks for joining us today. My name is Toby Hand. I'm the Vice President of SGA at the Lake Nona campus. And one reason I got vaccinated is to protect my grandmother who's currently living with us.
Hi, everybody. My name is Miranda Rubelitis. I'm the SGA president on Osceola campus, and I got vaccinated because my husband works with the public and literally meets hundreds of people a day. So we wanted to protect ourselves and those people.
Hi, everyone. My name is Ella Beatty, and I'm the Vice President of SGA for Osceola campus. And I got vaccinated because I wanted to keep my friends and my family safe.
Good afternoon, everyone. So my name is Isui Sopan. I'm the SGA president for the West Campus, and I as well got vaccinated to keep my family and friends safe.
Good morning, ladies and gentlemen. My name is Tajon Streets, and I'm the SGA Vice President here on the West Campus, and I got vaccinated to protect my community.
Good afternoon, everyone. My name is Pfizer Ahmed, and I'm SGA Winter Park Campus President. I got vaccinated because to save people around me and myself, mostly because at that time my parents were here and they both are 65 above. So that was important.
Hello, everybody. My name is Nomar Leva. I'm the Vice President for the Downtown Campus. I got vaccinated just to enjoy the freedoms that come along with it.
And we have a couple of guidelines that Ms. Pfizer will share with us.
Sure. So I would like to highlight the agenda of today's town hall. First, our guest speakers, Dr. Rita Luther, Dr. Gandhi Pierre-Louis, and Ms. Jessie Reed will be talking about the importance of vaccination. Please keep your microphones on mute so there is no disturbance during the presentations. The question and answer forum will be open at the end of the questions. If you have any questions, please raise your hand using the raise hand button. However, if you have any questions before that, you can post the questions in the chat. Olivia and Abigail will be monitoring the questions and professors will answer them after during the question and answer session. Thank you.
All right, I would like to introduce to you our speakers today. We have Dr. Rita Luther, who received a Bachelor of Science degree in microbiology from the University of Montana. Subsequently, Dr. Luther received a PhD in microbiology from the University of Alabama at Birmingham and completed her postdoctoral training at Sanford Burnham Previs Medical Discovery Institute in Orlando, Florida. Dr. Luther is currently a professor of biology at Valencia College, where she teaches both general biology and microbiology. In addition to that, she trains students and is the undergraduate research and serving as principal investigator of Vector. Dr. Gandhi Pierre-Louis, our second speaker, holds a Bachelor's of Science degree in biology with a concentration in cell and molecular biology from the State University of New York at Binghamton and a PhD degree in developmental biology from Stanford University. After completing his studies at Stanford, he trained as a postdoctoral research fellow at Emory University in the Department of Cell Biology. Since 2017, Dr. Pierre-Louis has been a faculty member at Valencia College, East Campus, and has taught numerous courses, including general biology and human anatomy and physiology. Professors, thank you for joining us today and for your passion for this important topic. We look forward to your informative presentations.
Thank you, Miranda and the rest of SGA. Can you guys see my screen, the welcome screen?
Yes, we can.
All right, well, thank you again for organizing such an important event. It's a pleasure to be here. I know Dr. Luther and I both share the same sentiments. So we were asked to present a little bit of background of particularly SARS-CoV-2 variants. So that's would be the focus of my talks, as well as a little bit of information about some vaccines. And that will be the focus of Dr. Luther's talk. I just wanted to provide a brief disclaimer before I begin, which is that Dr. Luther and I are not actively conducting research on COVID-19 or the virus that causes COVID-19. The information presented here were accessed by information that's publicly available. This presentation is for educational purposes only. We are not qualified to provide medical advice. Okay. So with that, I'll start with my brief presentation on SARS-CoV-2 variants and why they matter. So before I begin into the COVID variants, I just wanted to reiterate and remind folks about SARS-CoV-2, right, which is the virus, which is a novel coronavirus discovered in 2019, and is the virus that leads to COVID-19, right? So the virus is called SARS-CoV-2. And this initiated, long and heavy conversation when it was discovered that there was a zoonotic pathogenic event, which meant that the virus jumped from an animal to be able to infect humans. And that, of course, we know translates to a cascade of events because SARS-CoV-2 is highly transmissible between humans. It's just primarily via respiratory droplets. And often leads to lower respiratory infections, which means it could go and infect our lungs, essentially, right? But in addition to that, the virus specifically infects cells that have this receptor known as ACE2, so we'll talk a little bit about that in our talks. And some of these cells include cells of the lungs, blood cells, cells in the nervous system, and many more, okay? So it can cause a systematic issue. As you may know, in severe cases, infected individuals may produce an overactive immune response that leads to a condition known as acute respiratory distress syndrome, or ARDS. And that's essentially renders the lung unfunctional, and therefore, a person may need that individual may need to be on a respirator and has severe disease, and unfortunately may succumb to that disease. Okay, so that is SARS-CoV-2, and now let's begin our talk on the variants. But before I go into defining what variants are, I need to talk to you a little bit about how this virus or what this virus does when it enters our cells in our body, okay? So as I sort of mentioned before, the virus, SARS-CoV-2, can infect cells that has this receptor known as ACE2. The virus uses this binding between one of its proteins, known as a spike protein, to this receptor ACE2 to enter cells. And one thing you may know about viruses, one of the reasons why it must infect individuals or organisms is that it's unable to replicate itself. So what it does is it will infect cells, thereby and therefore hijacking the cellular machinery to replicate itself. So one of the things you may know is that SARS-CoV-2 is an RNA virus. That's its genetic material, and it will insert the genetic material inside living cells, and these cells unknowingly will start to process that RNA and undergo a process known as translation, which is taking that information and making proteins, in this case, viral proteins that the virus needs to replicate itself. And one of the viral proteins I will mention is a protein known as RNA-dependent RNA polymerase, which can also be used to replicate the RNA material that's going to get packaged into new viruses, okay, for replication. But one thing we need to understand about genetic replication in all cases is that it's oftentimes imperfect. So sometimes through the process of replicating genetic material, changes, also known as mutation, arise, leading to new versions of the organism, or in this case, virus. Because you may also know that genetic material encodes for components that is going to make up this virus, so proteins, et cetera. Okay. And so what does that mean, and how does that relate to SARS-CoV-2 variants? Well, what happens is, if you could imagine that this little cartoon of a virus here is the original SARS-CoV-2 virus that we detected back in 2019. And if you could imagine that this virus successfully infected a human and used that process to replicate itself, it will continue to, again, replicate within the individual and eventually transmit more viral particles into a new host. And it will just continue the process of replicating. But again, as I mentioned, through the process of genetic replication in particular, these viruses starts to accumulate mutations, so leading to changes and creating essentially different versions of these viruses. And so you can create variants, which I'm color coding here. So you can imagine blue is the original, but now you have these different versions, okay? But again, the process keeps repeating, right? So these variants are in the community, they go and infect different hosts, and they replicate and they spread. And you can start to see, in some cases, some of these variants starts to increase in number, right? So where you just maybe in this generation only saw a handful, now you're seeing a twofold number of variants, and that can continue to propagate. And there's also instances where some variants form and are not successful. But there are some cases where you may have different versions of the virus that's actually even more capable of infecting or being transmitted to a different host than, say, the original virus. And of course, we know that seems to be the case with the Delta variant, which is the latest variant of concern. So how can we stop variants from forming, right? So because as we see in the case of Delta variant, this is causing a lot of havoc in our society, where we thought we would be over this hurdle and things can get back to normal by now, but unfortunately, we still have to be quite careful. Right. And so how we can stop the spread of new variants that may be even more dangerous to Delta, we don't know, is to get to herd immunity. Right. And what is herd immunity? Well, I think this cartoon illustrates it nicely. So imagine if you have a population of people where the vast majority of people are vaccinated. Maybe you have a handful of people who are unvaccinated here in white and are definitely susceptible to being infected by a disease that may encounter. Well, these individuals can be, maybe they're unable to be vaccinated, they're due to their an allergy or something, or maybe they're too young, as we are currently seeing in our population, because the vaccine's only approved for a certain age. Well, if the people who can get vaccinated are vaccinated, they're kind of shielding the people who can't be vaccinated, so that if, again, the virus starts to enter this community here, it's unable to really propagate because it can't really infect very well or get transmitted. And so these people can be safe, right? So again, herd immunity, it will help us limit the spread. And again, one of the best ways to get to herd immunity is through vaccinations. And so with that, I will hand it over to Dr. Luther, who will tell us a little bit more about the vaccines. Thank you.
Thank you, Dr. Pierre-Louis, for that preliminary information on SARS-CoV-2. And thank you, Miranda, for the introduction, and as well as the disclaimer for the fact that I am not a medical professional and I'm not actively in research and just relaying information. Okay. Let me share my presentation here. And there we go. So what I'd like to talk about is what are these vaccines that are currently available in the United States made of, how do they work? So I'd like to provide some information about those components. And so that's what we'll talk about here in this presentation. So just to start off, we want to understand some basics of biology. Maybe you haven't seen this in a while, maybe you haven't seen it, but we want to start off by understanding how proteins are made in the cell. And I'm mainly focusing on cells like ours, which are eukaryotic cells. And if you remember, these cells have a nucleus. And in order to make proteins, these proteins are made through an original code, DNA, that's contained within the nucleus. And this provides a set of instructions to make that protein. And it's done so with this intermediate step of making a messenger RNA that conveys that code. And that part is done in the nucleus, which is then going to leave the nucleus and go to the cytoplasm, where then the protein can be generated with the help of a ribosome. And so we have that understanding, and we'll come back to some of those concepts as we look at how these vaccines are made and how they work. And as maybe you know, maybe you don't remember, when we talk about living organisms, the genetic material that serves as that code is DNA, and of course the intermediate is RNA. When we talk about viruses can sometimes have an RNA genome or a DNA genome. And as Dr. Pierre-Louis mentioned, SARS-CoV-2 has an RNA genome. And if you are familiar with DNA and RNA, there are one-letter symbols that represent those little notches, those little rungs of the ladder in DNA. And those are the nitrogenous bases, the A, T, Gs, and Cs. And in RNA, instead, you'll have A, U, Gs, and Cs. And those, what we call nucleotides, they comprise that genetic material. And what you're looking at here for SARS-CoV-2 is an image from a publication by Dongwan Kim. And it shows here at the top, there is a scale representing the number of those nucleotides. So the genome for SARS-CoV-2 is roughly 30,000 bases long. And the image below this shows a diagram of the gene regions. So there's several gene regions. And the main one we're going to focus on is this S gene. So this codes for the S protein, or the S glycoprotein. And that is shown here on the structure of the virus. And so this gene right here that's in the genome will provide a set of instructions to make the protein the S protein. And then it will be placed here in that part of the virus. This S protein, as Dr. Pierre-Louis mentioned, is a protein that's involved in the entry of the virus that we understand now into the cells that it infects. And you'll also see other features of the virus structure here. You've got some other proteins that are also encoded in its genome that are on the surface. And then it also has this envelope region in gray, which actually comes from the host cell that it infects. And then inside you have the genome that's surrounded by the yellow proteins, which are the capsid proteins. So that's kind of the general structure of the virus and where the S protein is positioned. Okay. And so this picture here is meant to just point out that when you're looking at this virus here, and here's your S protein on the SARS-CoV-2 virus, that before it has made contact with host cell surface receptors like ACE2 and this protease over here, this TMPRS22, before it has done that, it's in a structural state known as the pre-fusion state. So it hasn't started to fuse in and undergo any modifications as a result of entry into the cell. And that phrase is getting important when we talk about the vaccines, pre-fusion state. Okay, so again, over here on the left is just my little homemade picture of SARS-CoV-2. And on the right here is a homemade picture of representing what the vaccines would look like at the molecular level. So on the left side here, we have an example of what the Pfizer and Moderna vaccines would look like. Really rough sketch there. But inside this structure, you have this little red looking string here, and that represents an mRNA or messenger RNA that encodes for the S protein that would be on the surface of the virus. And then these blue spheres represent what's called a lipid nanoparticle. And the lipid nanoparticle is an important delivery system to get the RNA into a cell. So any type of nucleic acid being delivered into a cell, in this case, to be used as a vaccine, it needs some kind of carrier, some sort of deliverer. And for the RNA vaccines, that is going to be the lipid nanoparticle. For the Johnson & Johnson vaccine, this is, instead of being a lipid nanoparticle with an RNA strand inside, it will have instead comprise of an adenoviral vector. Okay, so what this is, adenovirus is a different kind of virus. And that's what the sort of red structure looks like here. And this virus is engineered specifically to not be able to replicate. So once this structure has entered into the cell, it's not going to create more of itself. Okay. Now inside of this, since adenovirus is a DNA virus, we'll have double-stranded DNA in here. And this little red sequence region here would represent the S gene. So that's what is in the genome of that adenoviral vector that is part of the Johnson & Johnson vaccine. And so both of these vaccines, the goal is the same, to get this nucleic acid, either the RNA or the DNA, that have the S gene into the cell so that those nucleic acid sequences can then direct the cell to make the S protein, okay? And so if we then, once that's done, then what we want to happen is for the immune system to see that S protein as, wait a minute, you're not supposed to be here, and then start to have a response to it, okay? Now, one of the things we have to consider about delivering a strand of RNA, I'm just gonna focus in a little bit on the RNA vaccines here, is that RNA is very unstable by itself. And so there are some considerations to think about for this that will lead us into why it's been developed the way it has, as having a lipid nanoparticle and a few other modifications. But RNA is unstable, once it enters into the body, there can be enzymes in the blood, in the cell, that can destroy the RNA. Other cells can just destroy, just anything that enters in that's not native, can be filtered out by the kidney. So it's not very stable. It also has a negative charge, and it is a larger molecule. So getting it across a cell membrane by itself is kind of a challenge. So it needs something to help it get inside the cell. Okay. So in order to address this, the researchers that have developed these vaccines have allowed for modifications in the RNA sequence, such that for, first of all, here in the first bullet, they have changed a couple of bases in the RNA so that there are two proline amino acids side by side that will allow for that S protein to remain in that pre-fusion state. Okay, remember we mentioned that before on the previous slide. And so that pre-fusion state, what they're trying to do there is they're trying to keep that S protein in a conformation that the immune system can more optimally view it and respond to it better, to provide better immune protection. In addition, some of the changes in the RNA help to stabilize the RNA so that you can then deliver it into the cell and also produce protein. And they also made some changes to decrease the immunogenicity that would cause like a nefarious immune response, so something that would not be good. So, so they've made certain modifications to prevent that, but still allow for protein production and have a response to that protein by the immune system. Also, the use of lipid nanoparticle, which there was research on this prior to the pandemic, where studies were done to understand, what components to put into a lipid nanoparticle. And in the case of the RNA vaccines now, for Moderna and Pfizer, they have these lipids here on the bottom of the slide here. Those are the lipids that are in the lipid nanoparticle. And their goal is to, help stabilize that RNA, help get it into the cell, and deliver it so that the protein can be produced. And one of the things that would be the hope for delivery of the lipid nanoparticle and the RNA is that it would get inside a special cell known as an antigen presenting cell. Okay. So just to explain a little bit about the processing of the vaccine once it gets inside the cell. And I just have a picture here from a publication that shows an example of, for the messenger RNA vaccines, the goal here is to deliver this messenger RNA vaccine inside that antigen presenting cell. And just to kind of give the really fast rundown of the immune response, the goal here with the immune response is if you have a pathogen that's infecting you, the immune response's goal is to get rid of that pathogen and remember it, okay? And that remembering is part of your acquired immunity. And so once this vaccine has been delivered into the cell, the goal is then to release that messenger RNA, make the S protein, and then the S protein can then be chopped up into this process down here with this proteasome. And because this is a special cell, it's called an antigen presenting cell, what it does is when it chops up this protein, this S protein, into these little pieces, they're called antigens, it will then present it on its cell surface, okay? And think of this as kind of like now, it's sort of like a display counter at a jewelry store or a big department store. And the antigen presenting cell's job is to basically rip apart these pieces of the pathogen and present them. Your other arm of the immune response, there's of course many arms here that I haven't really mentioned, but this other arm over here on the right includes what's called the third line of defense. And these are very specialized cells. And, what they do is they react very specifically to a specific piece of the pathogen. So it's very much like a specific identity that they're recognizing so they can hone in on that pathogen specifically. And so you'll notice that on the T helper cell here, there's a special receptor. It's also one on the cytotoxic T cell. And they can recognize those little pieces that are presented. But, we have a lot of these T cells and their T cell receptors can recognize different pieces. And so what happens is that these antigen presenting cells, they present these little antigen pieces. And let's say we have one T helper cell comes along and it sort of samples or taste tests that little antigen piece to see, do I recognize you? And if it doesn't, it just keeps moving. And then it'll go to the next one. Do I recognize you? No. Okay, keep going. And then once it recognizes the one that it recognizes that it's specified for, it will lock on, it will activate that T cell, and that T cell will begin dividing to make more of itself, and it will start sending out signals. And one of the things it can do is it can activate a B cell. And the B cell's job is to make antibodies. And the goal of antibodies is to flag the pathogen to get rid of it. So again, recapping the immune response, pathogen invades, we want to send signals to let B cells and T cells know something's up. And then we hopefully, in some cases here, we activate our B cells to make those antibodies. They flag the pathogen is destroyed, okay? We also have cytotoxic T cells that can kill virally infected cells directly. So we have kind of a multi-pronged approach at addressing the pathogen. And so the goal here in producing this S protein in the cell is to give something to your immune response to respond to. And when it does this, initially you'll have a lot of B cells and T cells that are proliferating, dividing very rapidly and responding. And for the most part, most of those cells are going to operate in the here and now and work to get rid of that pathogen. But once you start feeling better, most of those cells are going to die. But there's going to be a small percentage of them that will stick around. And those are your memory B cells and T cells. And they will remember that pathogen. So the next time you encounter that pathogen, they will be able to respond faster, more robustly, and you may not even notice that you encountered that pathogen. So it helps to protect you, and it helps to protect the amount of the virus that could be proliferating, because it's knocking it out earlier and faster. That's the idea. Okay. So so just to kind of show you some of the ingredients, this is available on the CDC website for all 3 vaccines. The active ingredient up here is of course, the RNA component for the Moderna and Pfizer vaccines, and then for Johnson & Johnson, it's the adenoviral vector. And then you can see the inactive ingredients here. The one thing I'll point out, which is also stated on the CDC website, is that these molecules with the red asterisk are potentially molecules that if you have an allergy to them, then that's something you'll want to talk to a medical professional about. And then in this slide here, it just shows you that for the clinical trials, the efficacy that was determined for each of these vaccines. And this is comparing individuals in the trials that either received the vaccine compared to those that received placebo. Okay, so you can see 95% for the Pfizer vaccine, 94.1% for Moderna, and 66.3% for Johnson & Johnson. I, of course, have not broken this down into all of the different breakdowns as far as, how does it affect this group or hospitalizations, et cetera. But these are just the basic statistics that are provided. And then for the eligibility for the Pfizer vaccine, this vaccine is FDA approved in the United States for 16 years and up. And for 12 to 15 years of age, it has an emergency use authorization, which is basically, and maybe others can shed light on this too, but basically this is, when there's an emergency and there's a need for something and there's a certain amount of data that can support the use of it, then it is able to be used, but there would be more data needed, more testing needed for approval. For Moderna and the Johnson & Johnson, it has an emergency use authorization for 18 years and up, okay? And then finally, regarding the vaccine booster, as you may have heard, soon there will be a booster available. And just to point out that the booster that you may have heard about, this has an emergency use authorization for the mRNA vaccines. So if you have previously had one of the mRNA vaccines administered to you, then if you're wanting to get the booster shot, then you would be able to do that 8 months beyond your second dose. And I believe those, I know it's within, this month that they're going to be available. And then for the Johnson & Johnson vaccine, there isn't sufficient evidence to have any recommendation for that at this time. But with that, I'll conclude there with my talk. And if you have any questions, please let me know. And if you'd like to segue into your next phase of the event, please go ahead, Miranda.
Hi, everyone. So thank you, professors, for your time. So we've been collecting questions from our various social media platforms over the past few weeks. So as you all start putting questions in the chat, I'll go ahead and pull one from there. So Dr. Pierre-Louis, based on your knowledge, what would you recommend as the best vaccine for the younger generation going to college, especially those on campus?
So that question is a medical question. I cannot suggest any particular vaccine. I don't know what your particular anyone's particular situations is. So you will definitely would have to contact your medical professional. They'll be the one to best tell you if you have questions about which vaccine you're eligible to take. You ask them. But as far as I know, the 3 vaccines that are at least under emergency use authorization are safe and effective. So oftentimes we're told to just take whatever it's available to you.
Thank you. Our second question is for Ms. Reed. How did research advances through the 90s and later published in 2005 about messenger RNA or mRNA help Pfizer and Moderna create a vaccine quickly?
Was that for me? Just want to make sure.
If either one could answer, we're open to anyone. So Professor Luther or Ms. Reed.
Oh.
Yeah, so I think, Professor Luther did a great job describing the difference in vaccines and really understanding that the use of the mRNA allowed for some improved efficiency in developing a vaccine that would help to build immunity a little bit more quickly. Historically, what had to be done was that those protein spikes had to be developed in a laboratory, and through the use of the messenger RNA, it allowed the body to develop their own immunity, which is really what helped to benefit in the fight against COVID-19. But I'll also kick it back to Professor Luther as well.
I guess I could probably add a little bit. I know from my reading that there was definitely prior studies in the lipid nanoparticle to understand how to use that as a vehicle. There were also prior studies with respect to some of those modifications to the RNA to stabilize and lower the immunogenicity. So, and just even the use of RNA as a therapeutic that, has started, pretty much, I believe, in the 90s and has continued forward. So research using RNA as some sort of intervention is not new. It is, it's been going on for quite some time now.
So we have another question in the chat, and the person is asking for more clarification on mRNA and what it really means and whether or not it does something to the cell specifically, like if it changes anything.
I could take that at first. And right. So, and I think Dr. Luther went into this a little bit more in her earlier presentation. But no, RNA does not change at least the genetic makeup of the host that acquires it, right? So keep in mind, right, as we mentioned, the virus SARS-CoV-2 is an RNA virus. So even without the vaccine, it would have injected its RNA into the cell. And it just uses that the RNA is sort of a template that certain components in the cell can use, sort of a blueprint that the cell can use to create protein, right? And that protein can be used for different things. So whether or not, so in the case of the vaccine, the RNA vaccines, what it does is, especially the RNA vaccines, right? So once we are injected into those, the RNA goes into our cells, and our cells will see that RNA just like any other RNA that's constantly being produced in our cells. And we'll use that to make a specific protein, in this case, the S protein, so that our body learns, once the body makes this S protein, and the S protein will go on the surface of the cells that injected the RNA, the body cells in the immune system will see that S protein as something foreign, and it will react against that foreign that antigen to create antibodies against it. And this is what we want, right? We want to be able to essentially safely train our immune system to learn that something is foreign, right? Because so that the next time, because that S protein by itself can't do any damage to our cells. But now your immune system recognizes S protein as something foreign, and it will build defenses around that, so that when you do actually come into contact or infected with an actual coronavirus, then your immune system will say, oh, I see that foreign S protein again. Let me attack anything that I see with S protein and destroy it. Okay, so I don't know if the others want to add anything else.
Yeah, I just wanted to add, and also realize that, so if you, for the vaccine, you're getting introduced that one protein into your cell. If somebody gets the infection, you're getting the whole virus and all the parts, and your immune system is going to be responding to all of that. And for some, maybe that works out fine, and maybe there's not that many symptoms. And for others, it might be more severe. And we're still trying to understand what makes it that some are more susceptible to severe infection versus others, especially as we have new variants. So in getting a vaccine like this, you're getting subjected to a small piece of the pathogen, so that you're priming your immune system to remember at least that little piece, and that gives you a little bit of a head start the next time, or if you do encounter it again, or ever at all, right?
Okay, so our next question is, are the cases in Florida really rising as fast as the news says? And does this correlate with breakthrough infections?
Yeah, I can take that one. And it is accurate that the cases in Florida are in a period of rapid increase. And if you look at hospitalizations, our confirmed current hospitalizations related to COVID-19 total pretty close to about 15,000. And when you look at that, when we talked about the importance of vaccination and allowing your body to build an immunity so that it can respond appropriately, about 90% of the people who are hospitalized are unvaccinated. So having that vaccine has a significant impact on your ability to either avoid illness or to avoid serious illness from the COVID-19 virus and many of the variants that we're experiencing now. But the numbers are rapidly, have been on a rapid increase. It seems that, at least for the hospitalization standpoint, that we've plateaued a little bit, but we still have a very high volume of people that are COVID positive in the hospitals, and more importantly, that have COVID-19 are in our intensive care units.
Our next question is, how is the COVID vaccine made so quickly compared to every other type of vaccine? New technologies, or was it just the emergency of the pandemic? And does that affect the safeness of it?
I'll just chime in. So it's a combination, right? So certainly this is one of the first uses of the RNA vaccines publicly. I mean, again, nanolipid particles were being explored to deliver other things clinically. So there was that new technology, and there was, right, this huge demand that the world was going through this pandemic, so it really made everyone put in gear, working 24/7 to solve this problem, to create a vaccine. So we know, right, that one of the better tools to curb a pandemic is to get people vaccinated so that we can acquire herd immunity. So there was there was a strong motivation plus new technology that made us, again, in record speed, be able to have these vaccines available. So please take advantage.
Okay, so our last question is, when deciding what vaccine to get, are the ingredients the only factor we should be looking at? this question is open to any of the professors or Miss Reed, who is open.
So can you repeat the question?
When deciding what vaccine to get, are the ingredients the only factor we should be looking at?
So I'll quickly, again, I wouldn't worry about the ingredients because, right, so these vaccines have gone through rigorous testing clinically to show that it is effective. Again, and you do want to talk to your medical professional, I guess they would know, again, in case you do have an allergy against one of the ingredients, they would be the ones to tell you, okay, maybe you should take this one instead of that one. But other than that, yeah, that's all I would have to say about that.
If I could chime in, I think it's important too to look at the efficacy of the vaccine. And what we do know is that with the mRNA vaccines, which are both the Pfizer and the Moderna, is that they are about 94 to 95% effective against the COVID viruses versus the Johnson & Johnson, which is still fairly effective and known to be as mutually effective against some of the more deadlier variants of COVID, but around 66% efficacy. So really the recommendation and what we are administering most often in the healthcare setting is the Pfizer and the Moderna.
Thank you so much to our speakers and our guests who are helping answer questions from Orlando Health. I want to mention really quickly, before we do our closing, don't leave just yet. To qualify for the Apple AirPod Pro giveaway, you will need to fill out the survey. That's going to automatically pop up when you close this Zoom session. And you'll be notified through your student email if you win the giveaway. And also we have a message from Dr. Plinsky, our college president, about something exciting that the college is doing, and you won't want to miss it.
Students, I wanted to share the news that if you're vaccinated, you could be eligible to win one of 100 prizes. We're going to have a drawing in both October and November, and if your name is drawn and you are fully vaccinated, you could be eligible to win prizes like a MacBook Pro, an iPad Pro, or even a year's worth of tuition. Please get vaccinated. It's the best way to protect yourself, your family, and our community.
Hello, guys. First of all, I just want to say thank you guys for coming out to this awesome vaccine town hall that we and the SGA members have posted. Well, we do have a couple of little bit more information for you guys. So as Dr. Polinsky said, we are having a Vaccinated Valencia event from our community organization. And so some of the prizes are, you could get 1 year free of tuition, you have a MacBook Pro or Apple AirPods. You have to be fully vaccinated in order to get it. So you must show your proof of vaccination in order to achieve one of these great prizes. Right on the screen right here, for those of you guys who haven't gotten vaccinated or want to get vaccinated, we have a QR code for the locations to get vaccinated. Also, if you are not comfortable with being on campus and getting vaccinated as well, your local pharmacies and your doctor's office, they have the vaccines. I personally got mine from Walmart. So all you have to do is just fill out a couple of documents and you'll be able to get your most likely Pfizer or Moderna vaccines. On here, we have the contact information for Dr. Luther and Dr. Pierre-Louis, as well as our SGA leaders. And if you have any questions or you guys want to know where you could get your vaccines, or maybe you guys didn't catch the information that we presented, please feel free to email us. This is by campus. So if you guys have any more questions or information that you want to ask or need a direction to be pointed, you can contact all of the SGA members and our two professors. Just to reiterate the whole point of this event, over 65,000 Americans have died from COVID-19, and most post the vaccine creation have been over 90% have not been vaccinated. At the end of the day, in order for us to live and be able to push through to a new life, we must all be vaccinated. So on behalf of Valencia College and the college-wide student government organization, let us protect our community by being vaccinated Valencians. Thank you guys for coming. And if you guys want to follow us for more events, please go to our social media, studentdevelopment.sbw on Instagram for more events or any more information on COVID. Thank you guys very much and have a good day.
Once again, thank you to our speakers, Dr. Luther, Dr. Pierre-Louis, and representatives from Orlando Health, Mrs. Reed. Also, don't forget, Valencia has some great things coming up. A year of tuition, that's fantastic. A MacBook Pro, Apple AirPods, all those things are great. But of course, we do this mainly so that we can protect each other and so that we can be more on campus and things like that as well. Just some clarification on that. You're going to see some more things coming out about the Vaccinated Valencia campaign. You will not have to give up your information in order to qualify. Any student who is enrolled is qualified for the giveaway. Once names are drawn, there will be 2 different drawings. Then you will be asked at that time to provide proof of vaccination in order to receive the prize. Again, thank you all for coming. Thank you, my fellow SGA members and everyone here. Have a good day, everybody.
Great job, SGA. Thank you.